On behalf of the MGH Research Institute and my own Department of Radiology and the Martinez Center for Biomedical Imaging, it's a great pleasure to be here today to talk to my colleague and a good friend, Matt Rosen. I often describe him as my better looking and much smarter younger brother, but in fact, no relation, much to my deficit. In any case, Matt, greetings. Thanks for joining me today and the rest of our audience.
Thank you, Uncle Bruce.
Uh-oh. I can see the conflict of interest.
It's already out.
Coming in, right? So, Matt, before we get started, why don't you give the audience a little sense for where you're speaking from today?
Absolutely.
Might help orient people to who you are and what you're all about.
Great. Yeah. So I'm in Building 75 at the Martinez Center, which is just sort of spitting distance from the Tobin Bridge. Let me just give a little tour. So out the window, which you can't quite see, the Tobin Bridge is there. And my lab really looks like a physics lab. So there's tools and not a lot of kind of wet bench things, electronics. And I'll give you a little tour of this is the homemade ultra low field MRI scanner that we're looking at right now. And then I'll pull back and here's some of the control electronics. So, you know, more transistors and soldering and less pipettes than a typical kind of wet lab, I would say.
Yeah, this doesn't look like anything you'd see in the Simcus building,
for instance.
Yeah, so, you know, tell the audience a little bit about your background. You have training in physics?
That's right, yeah, I got my PhD in atomic physics. So I started graduate school in '92, and I was really focusing on so-called hyperpolarization techniques, which are ways to transfer angular momentum from places you have it, like a laser beam, into places you want it, like nuclear spins, for the purpose of really improving the signal of NMR experiments. And the kind of NMR experiments I was interested in doing back then were really about searches for new physics, looking for like...
I'm going to interrupt you. When you say NMR, is that at all related to MRI, the kind of things that we use to get our scanners?
Right. So NMR, of course, is the measurement technology behind magnetic resonance imaging, but it's also a thing on its own. It's essentially a measurement technology for detecting, processing nuclear spins. And in this case, I was studying gases like helium and xenon, and I was making them magnetic or hyperpolarized. It's kind of an unusual concept, a magnetic gas. But these are inert, noble gases that have nuclear spins, that is, spins in their nucleus that you can add angular momentum to and make them very, very magnetic. And I was using these spins to essentially look for new physics. You know, we think of there's 4 forces in the universe. Is there a 5th force? Are there interesting things the way gravity couples or pseudo-vector particles, these kinds of very esoteric physics concepts. And I was working on that stuff, like I said, 92, 93, 94.
And then, so how did you go from this very esoteric physics to medicine and biology? What was the connection there?
Yeah, so around 94, one of my advisors who was in radiology, although he wasn't my advisor at the time, Scott Swanson, said, you guys are making these magnetic gases, and MRI uses NMR to image things that are essentially magnetic or polarized in a magnetic field. Boy, I bet we could make some pretty pictures if you take your highly magnetic gases and we do some imaging with them. So that started a real change in my career. I set up a bunch of physics experiments with lasers and high temperature things and ultra high vacuum experiments. And we did some of the very first imaging of, in that case, hyperpolarized xenon, which was made through this physics technique, inhaled by living rats, and then we could see it in the brain and the other organs of the body. That really gave me a taste for the idea that, wow, you can use physics tools to really improve what's happening in bioengineering, bioimaging, and hopefully translational research.
It's interesting that your first experiments were looking at the brain. I understand you have a little bit of a personal connection to the brain.
Yes, I do have a brain.
Besides,
yes. So my father is just retired last year at age 85. He was a neurologist still is a neurologist, practiced for more than 60 years. Started the neurology residency program at Stony Brook and then moved to Purdue about almost 20 years ago and just retired. And it's interesting you bring that up, Bruce, because he's a clinician scientist before, from before there was really such a thing. I mean, he didn't get a PhD like is de rigueur now for clinician scientists, but he ran a research laboratory full of homemade electronics, doing EEG and later patch clamping and all of these kind of things, computer programming. And that was something I really loved. As a young kid, I'd come into his lab and turn all the dials on his grass stimulators and change the calibration on all these things. But I was really fascinated by laboratory work, and that was really motivated me to really want to do something in my life where I could, have tactile involvement with the kind of experiments I was doing.
Yeah. Now, I think when I first met you, it was at the Center for Astrophysics.
Right.
Tell us a little bit about that place.
Yeah, so I started a postdoc in 2001 at the Center for Astrophysics in Ron Walsworth's lab. And at that time, I had all of this hyperpolarized gas imaging experience under my belt. And I wanted to see, were there other interesting interdisciplinary approaches or applications for this technology? So at the Center for Astrophysics, I was working on the studies of granular porous media using, again, these same techniques. We were continuing our bioimaging work, looking mostly at the lung. But the connection to the Center for Astrophysics really comes down to the fact that atomic clocks, hydrogen masers, these types of systems are very, very closely related.
All the things that I love deeply.
Exactly. So that work, the long history of atomic physics, back to Norman Ramsey, is really about these ideas of state-selected atoms, first in molecular beams and then in cells. And so it was kind of a natural thing for me to take this technology doing, again, increasing the angular momentum of these gases or liquids or solids, depending on the application, and trying to do something interesting with it.
And when you, back from graduate school and then in the CFA, the Center for Astrophysics, what were you thinking about where the biological applications were going? And, when did that kind of transition in your own mind go from somebody who was, broadly interested in physics,
right.
From the universe, as broad as the universe, to somebody that was really increasingly focused on the biological side of things?
Yeah, that really comes from the relationship between NMR and hyperpolarization and imaging. So the work I did in grad school, we were using a standard animal MRI scanner, and we were showing that we could really improve the signal and get maybe more information and make different types of images using hyperpolarization. But it dawned on us that if we had all of this magnetic gas to do NMR, we could actually do MRI now without having a very high magnetic field. We could build a very, very simple, almost Earth's field, very low magnetic field MRI scanner, breathe in a huff of this, say, hyperpolarized helium, and then make beautiful images of the lungs with the person standing up or with the person laying down. And the reason it's so interesting is because by turning the magnetic field down, number one, you can build a very inexpensive machine. And number two, for our purposes, we were interested in studying how the heart-lung system is affected by orientation in the gravitational field. So around 2004, with the student Leo Tsai, who's now at BI as an interventional radiologist, we built an MRI scanner that operated at very, very low magnetic field solely for the purpose of imaging the lungs after you inhale this hyperpolarized magnetic gas.
And I think that was the system that I first saw when I came and visited you, in the basement
Exactly.
of the Center for Astrophysics. But then, what prompted you to then take those experiments and come to the MGH?
So I really enjoyed working in that basement laboratory at the CFA, but it was actually quite challenging to do human experiments for lots of reasons. The Center for Astrophysics wasn't super excited about having, human medical experiments happening on their premises. They just weren't equipped. They didn't have an IRB. It was quite challenging. And so after meeting you, it became clear that there was some space over here. Again, space in the Boston area, very rare. But there was space enough that we could move that scanner over here and actually have a real research program. Again, at the time, I was focusing on lung imaging. move it over here and expand that program to, instead of scanning one person or two people a year, we could do 5 or 6 a week if we wanted to. And so there was a Catalyst grant that allowed us to move the equipment.
Harvard Catalyst?
Harvard Catalyst grant allowed us to renovate the space over here, put power in it, and then move the equipment over. And that's how I started over here in 2009.
And what was the kind of collaborative environment like when you came to the MGH? I mean, obviously a different kind of environment than the Center for Astrophysics.
It was a real learning experience for me. You know, we're physicists, in the group I was in, and we know a lot about certain things. And we thought we knew a lot, or I'll talk about myself. I thought I knew a lot about MRI, contemporary MRI. It turns out I met people like Larry Wald, who's the world's expert in contemporary high performance MRI. And it was clear that the sequences that we use, the sort of programs we use to acquire the data were, they were sort of contemporary or on or around 1979. And there were definitely new ways, more contemporary ways to take MRI data, process that data, build our radio frequency hardware. There's just huge amounts of things that we just didn't know. And so, picking on Larry as one example, it was a real learning experience for me. And very unlike what I've known in most physics departments. Faculty here like to collaborate. Everybody talks to everybody else. We have this amazing culture, which of course of everybody being on everyone else's grants, everyone else actually contributing parts to what they know, again, all in the service of advancing patient care. So that kind of made me fall in love, actually.
And how about on the more biological and medical side? Did people, kind of immediately gravitate to this technology, or did it take a little while for people to see through the crazy physics side of things and begin to see the medical potential of this technology?
I would say over here, people recognize that right away. I know Jerry Ackerman was really enthusiastic about it. But I would say once we started doing brain imaging and focusing on neuroimaging, then everybody was excited. And I can tell that story if you're interested.
Yeah, of course.
Yeah. So my focus again was going to just be on hyperpolarized lung imaging at low magnetic field. And around 2010, I got a grant from the Army. The Army was really interested in traumatic brain injury at the time. In particular,
they still are. They
still are. But at that time, Congress, because of an act of Congress, was really pushing through CDMRP, the Congressionally Directed Medical Research Program, a real effort looking at closed head TBI, mild TBI. And the Army was well aware that MRI is the undisputed standard of care when it comes to non-invasive neuroimaging. But the folks at the Army were really interested in saying, we want to scan warfighters in theater, in Iraq, in Afghanistan. And it was very clear that if those soldiers had, let's say, undisclosed metallic shrapnel in their head and they went into a 3T magnet, it would not be great. It would not be safe. And so they funded me basically to have a kind of Manhattan project, which was, okay, we know if we want to have MRI in the field, outside of the controlled access radiology suite, it needs to be low magnetic field. It needs to be safe enough that an oxygen cylinder or shrapnel or a surgical instrument won't get sucked out of the hands of the people who are operating it. So given that as a sort of motivating force, we threw all the physics and all the electrical engineering and all the compute and all the things that we knew and plenty of things we didn't know in my lab to sort of enable that. And that's where I would say I really discovered in kind of a deep dive the vast expertise here at the Martino Center, because I could talk to chemists, I could talk to neurologists. We have great collaborations with Jen Kayada and some other people, these real clinician scientists about, oh, so tell me a little bit about brain injury. Tell me about the markers of brain injury. And I got to learn things like, oh, free radicals are endogenous free radicals are associated. And I said, okay, I'm a physicist, and frankly, this is a bit embarrassing, but I didn't know what a free radical actually was.
Yeah, tell us about that, because I was going to drive us in that direction. That was now physicist meets medicine meets chemistry.
Exactly. And when it was explained to me that, well, the cascade of events is as follows. So you have a concussive event, let's say, and so you have a focal injury. And that focal injury stops when the impact stops. But in the minutes to hours to days following that injury, that focal traumatic brain injury becomes a global injury because the body overproduces these free radicals, which cause edema, cause swelling, and can take a small injury and make it quite life-threatening, actually. And this is a dynamic process that would be very nice to be able to study non-invasively. So what is a free radical? Oh, it's an electron. It's an electron spin. And I know about nuclear spin. I know about electron spins. And so that's something that, again, I'm embarrassed to say that I didn't know. But once that connection was made, that there's this interesting overproduction of free radicals associated with injury, they are essentially electron spins, and we know how to do magnetic resonance imaging, we were able to develop in my lab technology to essentially image the presence or absence, or get contrast from the presence or absence of those electron spins associated with injury. So we did some really nice work. Again, a wonderful collaboration with Jen Kayada in neurology, looking at oxidative stress in rodent models of stroke as a proxy for that.
Yeah, amazing. So you went from imaging, helium and xenon gas in the lung to studying the brain and starting with perfusion, and then diving right down into the underlying, molecular changes that occurred during, acute injury.
So that's a really interesting point because of a couple of reasons. This kind of free radical imaging is a new thing. It's a thing that you can't do on a clinical scanner, a high field scanner. And why, right? I said that's electron spins. Electron spins have a precession frequency that is 660 times larger than the normal thing, the normal protons that we look at. Why
isn't large good?
Yeah, so large is good, but it means in order to interrogate, the way you interrogate the body, the way you get information out of the water molecules typically in MRI is you shine radio frequency energy onto the body. And that frequency is typically, let's call it 100 megahertz. And that's fine, that's totally fine. It goes through the body, it's harmless.
Like FM.
Exactly. But if you want to look at electron spins, now we're talking about 50 gigahertz, which is microwave. So that would mean, Mr. Jones was fine before, he went into the scanner, but now you've microwaved him. And so it's not great. Now, at very, very low magnetic field where we operate in my lab, and everything in my lab is in the millitesla regime. So a typical clinical scanner, 1.5 Tesla or 3 Tesla. And we are at
Tesla is like 10,000 times or 20,000 times the Earth's magnetic field. So
that's right.
Pretty big magnet.
Exactly. And we are 100 times the Earth's magnetic field in my lab. And so it's very, very safe. The sort of nominal feeling of it is like the magnets on your refrigerator. And so by turning the magnetic field down, it makes a lot of other things harder. The signal you generally see is smaller, but the frequency that you use to interrogate these free radicals also drops. Yeah. And so we're able to do electron imaging or free radical imaging at 100 megahertz, just like a conventional MRI scanner. So that really got me thinking, like, wow, this is interesting. You can build these machines cheaply, low field MRI, and there are places where you can get information that you can't get on a traditional scanner. That was
simply couldn't do with the fancy Siemens or GE scanners we have.
Absolutely. And so that was the moment for me. That was really the moment. And from that point on, I asked every single clinician scientist here at the MGH and at the Martino Center, so tell me a problem that you're working on, and like, what would you like? What kind of dynamic imaging process? What kind of biomarker thing? Like, what would really change care?
It's the first question I always ask if somebody comes by. It's like, what would you like to image? And then it seems like it's, our job, your job, to figure out how to do it.
Exactly. And there have been, of course, there's some misses. There are some things where I don't know, or it can't be done, or I'm not smart enough. But there have been some great things where, it's a language thing. Once the translation between the sort of pathology, neuroanatomy, neurophysiology, clinical medicine gets translated into chemistry and physics, so that we can have a conversation. It's like being at the UN and everyone has their microphones on. Lots of things can come out of that often we can address experimentally in my lab.
Yeah, let me show. I'm going to share a screen for 1 second, and I just want to share with the audience here. Can folks see that?
Yep.
You looking at what we're hoping to? Okay, so tell us about this picture.
Yeah, so that was in Popular Science. That is me standing in the scanner that I just showed at the beginning. It was actually shortly after I moved over here, and Popular Science was calling us the DoItYourself MRI, and you can see
you
that
literally did build this magnet by hand yourself.
Exactly. So the whole scanner was built by me and a graduate student, Leo Tsai, back in 2004. When I moved over here, 2009, I rebuilt some. We had learned a few things about imaging, and I'd rebuilt some of the key components. But that scanner is still the test bed system in my laboratory for everything we do at low magnetic field.
So let's wind forward. That article I'm looking at now, Popular Science, November 2010, so more than a decade ago. What's happened to that technology since then?
Right. Well, one of the very earliest conversations I had with you, Bruce, was, okay, that's great that you're doing the lungs, but how about the brain? You know, we're at the Martino Center. Can you do things with the brain? And I you know, it's what motivated me to apply for that Army money. And then, you know, we had a lot of money. It was my first very big grant, and we had to produce on it. So I hired amazing postdocs. We had great students, and within a few short years, we were able to demonstrate that it's actually possible to do, let's call it, pretty good imaging, pretty good traditional brain imaging at these extremely low, like 100 times higher than the Earth's magnetic field, in a very simple and cheap system. So that changed things a lot. That kind of became an existence proof for lots of people who are working in the space, because everyone wants to, not everyone, but there are lots of people who want to make magnetic resonance imaging affordable, portable, ubiquitous, and really change healthcare. And so we had, you know, once you have a glimpse, it's like if you see someone juggling, you know, 3 balls or 5 balls or 7 balls, you realize it is possible, and you'll work really hard to try to do it. But if you've never seen someone do it, it's impossible, right? So
until somebody shows you it can be done, and now suddenly it's very possible.
Exactly. And so that really took off for us. And so since that time, we've developed a lot of technology, again, on the compute side, on the physics side, on the electrical engineering side, on the AI side, and we've spun off some of these ideas as well into the commercial sector.
Tell us about you know, some of these spin-offs. One in particular I know has become you know, important on the clinical arena.
Yeah, so 7 years ago, I was approached by Jonathan Rothberg, who is a well-known entrepreneur in the biospace. Ion Torrent was his first real foray into this
like
space.
Gene chip kind of stuff.
The $50,000 gene sequencing tool. And then more recently, Butterfly, again, that's ultrasound on a chip. And so he reached out to me. He'd seen some of my work, and he said, could we build a $50,000 MRI?
This Popular Science article.
Exactly. Could we build a $50,000 MRI scanner? And of course, MRI scanners at the time, well, and today, they're still multi-million-dollar machines.
Yeah, the answer must be no.
And so I said, yes, in fact, we're doing it already. You know, because we had some really good results, and I know how much it costs to build this machine. And so Jonathan and I, with Ron Walsworth at Harvard, founded a company called Hyperfine to really say, okay, you know, I'm a physicist. The limits of what I know in terms of engineering are small. But if you're well capitalized, you have a great story, a great healthcare application, you can just get people who are incredibly skilled in the engineering space to take a deep dive and work on miniaturizing and reducing the cost of that technology. And so now, as you said, yes, Hyperfine sells a $50,000 portable MRI scanner that's used predominantly for neuroimaging. The MGH has 2 of them over in London.
I didn't hear about the second one. I know about the first one.
Yes. Taylor Kimberly has 2. They're very small. We published a paper in JAMA Neurology last year showing at Yale New Haven Hospital, in work led by Kevin Sheh, who's the director of neurocritical care, that you could actually image unconscious comatose COVID patients at their bedside, right at their bedside, without having to disconnect any ventilators or EKG or anything, without having to transport them into the radiology suite, and do reasonable neuroimaging right there to help with diagnosis of neurological deficit. Interestingly, in that case, these were unconscious patients, and so you couldn't do a normal neuro exam, and they were too sick to transport them to the regular radiology suite. So they were really able to, by virtue of portability and safety. Again, full circle really. Operation at low magnetic field means it could be right there in the neuro ICU at the bedside. You could get information that would advise treatment. So that's really like incredibly rewarding to see that slice of what we've been doing in my lab. Again, funded by the DoD back in 2010 with a goal of trauma, now getting out and being a real success story.
So, when we talk at the MGH, as we frequently do, about bench to bedside, in your case, it's lab bench, it's physics bench.
That's right. That's right.
From the Center for Astrophysics to literally providing bedside care. It's a completely amazing story.
Exactly.
Well, you and I could probably keep chatting and for hours, but
where did the time go? Right.
At the time go, exactly. We've managed, what do they say in car talk? You've managed to waste another hour. In this case, just a half an hour. Listen, frickin' frack, the Rosen brothers, talking. We're very glad for all of you that have listened in. I see more than 80 people are listening. That's fantastic. Reach out to Matt to learn more about his amazing work. It's quite impressive, lots of fun, and I thank you and the Research Institute in particular for the opportunity to have the chance to have this conversation. Matt, thanks so much for your time.
Yeah, and Bruce, I have to put an ad in for the Martino Center. Again, the Martino Center is this amazing place where you find scientists of all subspecialties, all working to advance healthcare, and it's an incredibly creative place. And check out the Martino Center website and reach out to all of us, not just me. It's an amazing place.
Excellent. Okay, well, with that, I guess we're done. Do I just shut this down or up? Avery Burns, thanks for everybody. Thanking the Rosen brothers. There we go.
Thanks a lot, Bruce, and thanks everybody.
See you all soon. Bye bye now.